Hi, my name is Montanna Cassel. I'm from the Department of Biology at Towson University, and today I'll be explaining the first experiment my group and I conducted in chemistry about enzyme activity. Here's my introduction. The proteolytic enzyme trypsin is necessary to all living organisms as it helps break down and digest proteins. It is produced in the human pancreas in its inactive state, trypsinogen, and once it enters the small intestines, it becomes active and transforms into trypsin. Similarly to all enzymes, trypsin can be denaturized, which means loss of biological activity. This was also one of the goals of our experiment, and in order to discover the optimal level of the enzyme activity with trypsin, the catalyzed enzyme, and azocasein, the substrate, my group used a range of different salt concentrations, also known as our buffers, between 0.0 NaCl to 4.8 NaCl. Our question for our hypothesis is, what is the optimal salt concentration for increased trypsin activity? And from what my group concluded from the first trial of the experiment is that our optimal salt concentration will be 0.0 moles of NaCl. The materials and methods are right here, and the materials we used were azocasein, our substrate, trypsin, our catalyzed enzyme, and 10% of TCA, along with our buffers, which I already said range from 0.0 NaCl to 4.8 NaCl, along with deionized water for our negative controls. So, each reaction consisted of 500 microliters of azocasein, the substrate, and 50 microliters of trypsin, the catalyzed enzyme. And each had 450 microliters of a different salt concentration, ranging right there. And at a pH level of 8, all of the reactions were incubated at 52 degrees Celsius. And after the incubation, we added 500 microliters of the 10% TCA to begin the process of denaturizing. We then centrifuged the reactions for 2 minutes at 12,000 RPM, separating the pellet from the supernatant. The pellet is the digested substrate, and the supernatant is the undigested substrate. The pellet is a kind of solid looking form at the bottom of your tube, and the supernatant is the liquid sitting at the top. The addition of 500 azocasein is a protein milk and is due to the sulfaldamide group within azocasein that allows for the reaction to be detected by the UV spectrometer, which observes the absorbency rate of the end result. The supernatant is placed in the spectrometer at 440 nanometers to be tested for the absorbency. The spectrometer is blinked prior to the first reaction being put in because it allows for a more accurate answer. And how we blinked it is we put deionized water in a little tube and we just put it in the machine. And so for every reaction, we did use a negative control because it allows us to identify whether or not azocasein is in place of in the absence of the enzyme activity site. My apologies. The trypsin was replaced by the deionized water, and we found for each reaction that our results were similar to each reaction's trial mean, demonstrating that azocasein was present in the empty enzyme activity site. Also, each reaction did have 3 trials. We do 3 trials for a more accurate answer and allowing the person or the observer to draw a better conclusion for the experiment. Also, 3 trials are done, which you will see in our results later, to find any errors that the experimenter has made along the way that could be fixed in a future upcoming experiment. The reaction temperature and pH level, so the reaction level again was the reaction temperature level was 52 degrees Celsius and the pH level was 8. And my group chose these based upon information gathered in the first experimental trial. And in the first trial, we tested the reactions at 37 degrees Celsius, which is the average human body temperature, as well as the average temperature for the active enzyme activity in the human body. Similarly to temperature, the normal pH level inside a human is around 7.35 to about 7.45, and so that is why we used 8 as our pH level. This allows for us to copy the conditions of the human body in a lab instead, and it gives us a better environment. It allows for the activity to be more precise as well. So throughout our experiment, before the incubation, all the reactions were the same color. They were about an orange yellowish. They all looked the same. After the incubation, they were more yellow, still looked the same, not a lot of variation of color. And after a centrifuge, as I said before, you see the pellet and you see the supernant, which is the liquid and the solid. Again, they're the same color. And when you put into the cuvette, which is the small little tube you put into the UV spectrometer, it is the same color to the naked eye as something more magnified. Our results here show that our hypothesis was correct, as our salt concentration was 0.0 moles of NaCl. The results chart below show that the standard deviation for 0.0 NaCl was at 0.01473, which was our lowest result, meaning that is the optimal level. And we perform standard deviation because it allows for a more accurate result rather than using the mean or the median of our reactions results. Also, a t-test over here was conducted to identify the significant differences between each experiment. So below, as I said, we did 3 trials for our experiment. For each reaction, my apologies, we have our mean, our negative control for each reaction, as well as our standard deviation. Our optimal level of 0.0 moles of NaCl had a standard deviation, as I said before, of 0.1473. 0.075 moles of NaCl had a standard deviation of 0.03175. 0.15 moles of NaCl was 0.03292, and for 0.3 moles of NaCl along with 0.6 moles of NaCl were slightly different compared to the other reaction, as both started out at a much lower reaction rate. The first trial showed for 0.3 moles was 0.595, and for 0.6 it was 0.570, and this can be a result of incorrectly pipetting, contamination of something, not wearing gloves, not checking our machines, putting something in for too long or at a wrong rate. It could be a number of things that my group would be more precautious with if we were to do this trial again. And as I said before, the optimal level is 0.0 moles of NaCl, but if we had not done standard deviation, my group would have had assumed that it was 0.6 moles of NaCl, because the mean is just as low as 0.0. And so here are T results, and for our T results here, we show that there's no significance between 0.0 moles of NaCl compared to 0.075 moles of NaCl, along with no significance difference in 0.05 and 0.15. Also, there is no significant difference in 0.15 moles of NaCl compared to 0.3 moles of NaCl. And lastly, there's no significant difference in 0.3 NaCl moles compared to 0.6 moles of NaCl. Overall, the hypothesis of the optimal salt concentration being 0.0 moles of NaCl was correct. The results of the standard deviation supported the hypothesis, as standard deviation is more specific than calculating the mean. And as I stated before, if I had not calculated the standard deviation, my group would have had assumed that the optimal level is 0.6 moles of NaCl. And It was also helpful that in our first trial of the experiment that we narrowed down our buffer concentrations from 0.0 to 4.8 down to 0.0 to 0.6 moles of NaCl. Although the rate of our salt concentration did increase and was pretty high, it could have been higher, and I discovered this by comparing all the group concentrations and buffers that they use, and I discovered that one group that used temperature, Virginia, Simi, and Tabby's group, reached a rate of 0.006658328 at 77 degrees Celsius, which means that temperature is the best buffer possible to find the most increased level of trypsin activity. Unfortunately, my group did not reach this, possibly due to contamination and those similar actions that I explained earlier that could have caused a fault in our results or a fault in some step along the way. A following question that was asked in our experiment was, would this procedure be a good way to grow bacteria? And yes, I do agree, as this is a good head weight. You have buffers, you can find your control you can find your control variables, your experimental variables. You have your buffers, which are your control variables already, and from the information that I learned and gathered throughout the process, it shows that at a higher temperature, you're going to receive more results and a better rate of growth. Thank you.